Okay, everyone, welcome to the second lecture for Bio 139 online. We're picking up where we left off last time. Previously, we introduced blood. We talked a little bit about just the general overview of blood. And this lecture is going to be talking about erythrocytes, red blood cells. So, if this were an in-person class, what I do is every class we start with attendance questions, and they're not for points. What I do is I ask these questions, everyone writes their answers down on a piece of paper and turns them in, and that's how I take attendance for the day. But it also helps everyone kind of get a feel for how well are they retaining the material or understanding the material, either from the previous day or just somewhere along the way so far. In order to kind of give you guys that same experience, I will start each lecture, now that we're past the first one, with some not really attendance questions. And these are questions just like I would ask in class, and you can use them to try to gauge where you are with your understanding at any point. So, today's questions are: what is normal hematocrit? And what specific component of blood contributes to colloid osmotic pressure, or COP? So what I like to ask you to do, now that we're online instead of in person, pause this and try to answer them before you get the correct answer. Okay, so hopefully you've tried to answer these on your own, but let's see what the correct answers are. First, what is normal hematocrit? Well, let's actually go a little before that and first ask, what is hematocrit? We have to know what that word means before we can talk about what the normal hematocrit is. And hematocrit, remember, that's the portion of whole blood made up of red blood cells, or erythrocytes, which is what we're going to be talking about today. And in the average healthy adult male, hematocrit is about 45%. 45 to 52 is what your book gives, but we can just say about 45% is considered normal for the average healthy adult male. Next, what specific component of blood contributes most to colloid osmotic pressure? Well, remember, colloid osmotic pressure is that pulling force. So stuff in the plasma pulling water towards itself, holding water in the blood so it doesn't all leak out at the capillaries. So which component of blood, specifically which component of plasma, contributes most? Well, that was that protein albumin. Albumin is a very large protein, and that's its primary function, is to hold water in the blood. All right, so those are the correct answers, and hopefully you are able to recall those and were able to answer those. If not, it doesn't really mean that you're far behind or anything like that, but it does kind of give you an idea of maybe I need to take notes a little more or pay a little bit more attention, and try something to help this information stick in my head. All right? Well, let's get rolling with new material for this lecture, and we're going to talk first about formed elements. All right, last time we talked about plasma. Plasma was the liquid portion of blood and the stuff that was dissolved in it. Formed elements are the solid parts of blood. So the things that we're going to be talking about pretty much for the rest of this chapter are formed elements. Now, there's different formed elements, and they all have something in common, and that is they are all cellular in origin. They are either cells or something that came from a cell but has changed a bit. So something unusual about red blood cells and platelets This is what I mean by something has changed with them. Red blood cells and platelets, they're not really cells. Red blood cells don't have a nucleus. They don't have any DNA. So we can't really call them cells. Platelets are actually pieces of cell cytoplasm. And we're going to talk about red blood cells and platelets both in depth. But for now, just know they started out as cells, but they are not cells from the point that they actually become red blood cells or platelets. Formed elements are mostly amitotic. Think about what that word means. It sounds like mitosis. Remember, mitosis was cell division. But when we see a at the front of a word, that means without. So amitotic means that these cells or former cells, they do not really go through mitosis. Red blood cells don't go through mitosis. Platelets don't go through mitosis. White blood cells, they are capable of going through mitosis, but really they only do it at very specific times. And we'll see later what those times are. But as a general rule, formed elements do not go through cell division, mitosis. And they are all made in the red bone marrow. Every formed element comes from red bone marrow. So today we're talking about erythrocytes, and that's what we see here. We saw them in this previous picture. Remember, we said these were like the little pink donut looking things. But here's kind of a cartoonized picture of it. This is from page 676 in your textbook. And here are some terms about erythrocytes. Now, erythrocytes, this is the proper word for a red blood cell. I don't ever like to throw science words at anyone without first, talking them through what those words actually mean. So up to this point, I've kind of used red blood cell. I've mentioned erythrocytes a time or two, but now we're actually going to start using the word erythrocyte a little more often. Erythrocytes are red blood cells. Erythro is a root word that means red. Cyte is a root word that means cell. So erythrocytes are red cells. And here are some properties of red blood cells or erythrocytes. They have a very high surface area to volume ratio. That just means for the amount of volume that an erythrocyte has, it's actually got a lot of surface area. It is biconcave, which really just means, concave means sunken in, but it's biconcave, meaning it's sunken in twice. Both sides are sunken in. Organelles. Well, think back to when you were learning about cells and all the organelles that they have. Erythrocytes don't have them. The nucleus and all of the other organelles are actually ejected after they have been made and just before they leave the bone marrow. So the organelles are missing. Now, that is why we have this biconcave shape. They start out shaped like a regular cell, kind of globular, and then when those organelles are kicked out, they cave in on themselves, getting this biconcave shape. And everything in our body is mostly water, but if we ignore the water part and we only look at everything else in an erythrocyte, 97% of that remaining stuff is hemoglobin. Okay? So, if you've had me before, you might be familiar with what I call thinking cap questions. And these are when you are filling out your outline and you see 4 or 5 asterisks in a row with nothing written next to them, that's a heads up that there's a thinking cap question coming. And these are kind of like those attendance questions that we just did. These are saying, hey, based on this stuff that we were just talking about, let's put it together and see, do you understand what we were just talking about? So here's our first thinking cap question of the semester. Why are those qualities, high surface area to volume ratio, biconcave, organelles are missing, and 97% of the non-water portion is hemoglobin. Why are those qualities important for an erythrocyte? And as kind of a hint, what is the job of an erythrocyte? See if you can answer this before I give the answer. So pause it and see if you know what the answer is. Okay, so here are those qualities. Let's think about why these things are important. Well, all of these qualities make it really, really efficient for these cells to carry a ton of oxygen. That's the job of an erythrocyte, is to carry oxygen to our tissues. High surface area to volume ratio. Well, the oxygen is carried inside, so the oxygen needs to be able to pass through. Well, the more surface there is, the more space there is for oxygen to move into the cell. So very high surface area to volume ratio makes it really efficient for oxygen to move in and out. Biconcave, well, the biconcave is why it has that high surface area to volume ratio. Organelles missing. Why would we want our organelles to be missing? Well, organelles use ATP. Where does ATP come from? The mitochondria use oxygen and a few other things to make ATP. So if we get rid of the organelles that need ATP, and we get rid of the organelles that make ATP using oxygen, there's no reason for this cell to need the oxygen that it's carrying. It's just basically like a trailer full of oxygen. It doesn't need the oxygen, it's just delivering it. And other than water, it's 97% hemoglobin. Hemoglobin is the protein that oxygen binds to. So pack this thing full of the protein that actually does the oxygen carrying. All of those things are really, really good at making the erythrocyte better at carrying oxygen. So here is our hemoglobin. This again is on page 676 of your textbook. And the function of hemoglobin is to carry oxygen. We'll see later in the semester it also carries carbon dioxide, but oxygen transport is the real main function of hemoglobin. And hemoglobin, it has a quaternary structure. Maybe you're familiar with that word from Bio 137, maybe not. But remember, when proteins are being made, first they have a bunch of amino acids that get stuck together, and we call that the primary structure of that protein, the order of amino acids. And then they folded up and coiled up, and we called that the secondary structure. And then it folded up to get kind of a three-dimensional shape, and we called that the tertiary structure, and then it was a protein. But sometimes more than one protein will stick together, and we call that quaternary structure. Quaternary structure just means it's a protein made of multiple proteins stuck together. In the case of hemoglobin, it's actually made of 4 separate proteins stuck together. 2 of them are exactly the same, and these are these gold pieces right here. 2 of them are exactly the same, and that's these kind of bluish purple pieces right here. The bluish purple are called the alpha subunits, and there's 2 of them. The gold ones are called the beta subunits, and there's 2 of them. And that's the actual protein part. But in the middle of each of those protein subunits is something that is not a protein, and this is called the heme pigment. That's what we see right here. And here is, if you're big on chemistry, I'm not, but if you like chemistry, this is the chemical structure of that heme pigment. I will not ask you to know this. And the important thing about the heme pigment is what's right smack in the middle is this little blue-green ball right there in the smack dab middle of every heme pigment. Look down here, Fe. Does anyone remember what Fe is? That's iron. Every alpha unit and every beta unit has a heme pigment, and every heme pigment has 1 iron atom. and the iron atom is what oxygen is attracted to. So one oxygen molecule will attach to the heme pigment, specifically to the iron atom, in each hemoglobin molecule. So how many oxygens can one hemoglobin molecule carry? 4. And there are millions of them in every erythrocyte. All right, well, in addition to carrying oxygen and to a smaller degree carbon dioxide, hemoglobin is also a buffer. Do you remember what a buffer is? A buffer is something that resists pH change. Now, later in the semester, when we talk about the respiratory system and when we talk about pH maintenance, we're going to see how hemoglobin actually functions as a buffer. Don't worry about it right now. Just know that it is a buffer and file that away, because we're going to come back to that when we get to respiratory and when we get to pH maintenance. Well, that's what the erythrocytes do, but how do we produce these red blood cells, these erythrocytes? Where are they made? Why are they made? Well, first let's look at a couple of terms. Hematopoiesis. Hematopoiesis. Crazy looking word, I know. When you see poiesis at the end of a word, that means formation of. Hemata is blood. So hematopoiesis is blood formation. That's what the red bone marrow does. It's making blood constantly. But there's actually different parts of hematopoiesis. So erythropoiesis, you might be able to piece this together now that we've seen some of these root words. Erythro, red, poiesis, formation. So erythropoiesis is a part of hematopoiesis, but erythropoiesis is specifically making red blood cells, making erythrocytes. All right, so where is this erythropoiesis taking place? It's taking place in the red bone marrow. Specifically, there are these certain stem cells called hematopoietic or hemopoietic stem cells. Different books will call them different things. I think our current textbook calls it hemopoietic stem cell. Some other textbooks say hematopoietic stem cell. They're the same thing. But these are the parts of the red bone marrow that actually make erythrocytes. So hemopoietic stem cells, or hematopoietic stem cells, also called hemocytoblast. Remember, blast is a word that means to build or to make. So these hematopoietic or hemopoietic stem cells, these hemocytoblasts, are the cells that actually make erythrocytes. So they are functioning in erythropoiesis. And remember, at the end, so once we've made these red blood cells, they're going to start to go through some changes, and at the very end, the organelles are ejected. And here's a kind of a step-by-step thing on page 677 of your textbook. Guess what? You don't have to know this. I do want you to know what you start with. Hemopoietic stem cell, hemocytoblast. I want you to know that there's some changes that happen. So what actually happens is the hemopoietic stem cell, like every stem cell in your body, it's constantly dividing. When it divides, it produces 2 new cells. One cell remains a stem cell. The other cell is going to be what goes through these changes. So if we're going to make red blood cells, we divide, we get 2 new cells. One of them remains a stem cell. One of them is what we see here, a colony-forming unit. You don't have to know that. It goes through some changes, it goes through some changes, it kicks out the nucleus and all the organelles, and now it's an erythrocyte, and that's what enters your bloodstream. So what do you need to know from this? The cell that you start with, that the nucleus and the organelles are ejected, and that you end with an erythrocyte. You do not need to know all of these intermediate steps. Well, that's how they're made, but why are they made? How is it controlled? How do we know when we need to make them and when we don't? Well, it's actually under control of a hormone called erythropoietin. Erythropoietin, or EPO. You might have heard about this. It's actually something that's really, really talked about either during the Olympics, during Tour de France, during a lot of big athletic events, because of something called blood doping. So erythropoietin is actually made in, of all places, your kidneys. Your kidneys detect when you need to make more red blood cells. And when they detect that you need to make more red blood cells, they release erythropoietin into the blood. That erythropoietin is carried to the red bone marrow, and it stimulates the red bone marrow to go through erythropoiesis. So how does that tie into blood doping? Well, it's not uncommon for certain athletes to inject erythropoietin. And if you inject erythropoietin, what's going to happen? It's going to cause you to make more red blood cells. Why would you want that? Well, if you have more red blood cells, then you can carry more oxygen. More oxygen, it gives you actually a performance advantage. Your muscles can work harder for longer. And it's really hard to detect because erythropoietin is something that is naturally in your body, so it doesn't really look out of place. The problem with that, see if you can think of what the problem might be. If you have more red blood cells, especially if you're intentionally making yourself make more red blood cells above what you really normally need, what's going to happen? Think about what we started this lecture with. Your hematocrit is going to go up. You're going to have more red blood cells. Your hematocrit goes up. In the previous lecture, what did we learn that happens when your hematocrit goes up? Your viscosity increases, so your blood gets more viscous. And when it gets more viscous, what does that cause? It causes your blood to have more resistance to flow. It's not going to flow through your blood vessels as easy because now it's thicker. And that causes your heart to work harder. And blood doping can often lead to heart failure because you are overtaxing your heart. So, erythropoietin is what stimulates erythropoiesis, causing more red blood cells. What are the stimuli? Why would we need to make more red blood cells? Well, something like low oxygen. If you have low oxygen in your blood, well, we want to make more red blood cells so that we can carry more oxygen. Low red blood cell count. If you don't have enough red blood cells, then you're not going to be carrying enough oxygen. And again, that will stimulate erythropoietin. Low hemoglobin count. Maybe you're not making enough hemoglobin. Well, make more red blood cells. More red blood cells, more hemoglobin. Testosterone. Testosterone actually stimulates erythropoietin to be released, which if you go back to the chart that was, let's see, on page 672, you'll see that in males, the normal hematocrit is 45 to 52, but in females, it's 37 to 48. So males have a higher hematocrit, probably because of the testosterone being higher in males. Now, it's not that males have testosterone and females don't. It's just males tend to have more testosterone. And lastly, I'm sorry if there was a glitch in the recording. My computer froze up on me, I think. But lastly, vitamin B12, folic acid, intrinsic factor, how are these related to erythrocyte production? Well, intrinsic factor is something made by the lining of the stomach. And intrinsic factor is actually needed to absorb vitamin B12 and folic acid from your diet. And if you are deficient in intrinsic factor, then you can't absorb vitamin B12, and that will lead to a disorder that we will talk about when we get to the various types of anemias. But just know that intrinsic factor is needed for vitamin B12 and folic acid, and vitamin B12, folic acid is needed for erythrocyte production. Erythrocyte homeostasis. So here's that word, homeostasis. We used it a lot in Bio 137. We're going to use it a lot in Bio 139. And here's our homeostatic feedback loop. And this is really just kind of an illustration of everything we just said. This is on page 679 of your textbook. And what we see here is hypoxemia. Well, hypo, that means low. Oxemia, something related to oxygen. Specifically, this is blood oxygen. So low blood oxygen, inadequate oxygen transport. Sensed by the kidneys. It's also sensed by the liver, but it's more the kidneys. And then the kidneys respond, or the liver responds, by releasing EPO, erythropoietin. Erythropoietin circulates in your blood until it gets to the red bone marrow. Red bone marrow. Now we're going to go through erythropoiesis, make more red blood cells, so your red blood cell count increases, your hematocrit increases. When you have more red blood cells, now we can carry more oxygen. So that's our homeostatic feedback loop. But red blood cells, erythrocytes, don't last very long. Some cells in our body will last practically as long as we do. Some cells will last, months or years. But red blood cells, they don't last very long at all. They only live about 120 days. And that's for a couple of reasons. One, they don't have the ability to repair themselves or to make things that the cell might need because they have no organelles. They have no nucleus and DNA. And when these red blood cells are circulating, that's actually really hard on cells. They're constantly bumping up against things, squeezing, bending, folding, and it does a lot of damage, but they can't repair that damage. So they last about 120 days. And after about that 120-day mark, they're destroyed in the liver, in the spleen, really primarily by the spleen, but the liver, the kidneys, a few other places, by macrophages. Remember macrophages from Bio 137? These are really big phagocytic cells. They like to eat things. So they're going to kind of break down these red blood cells and their contents. And like we saw a minute ago, these red blood cells, their contents, it's mostly hemoglobin. And honestly, that's the important thing that we're after. So while the things like the phospholipid bilayer are still in a red blood cell, we're going to see what happens to the hemoglobin once this red blood cell is broken down. Here, this picture is also on page 679 of your textbook. And the hemoglobin is actually going to be broken down into two parts. It's going to be broken down into heme and globin. So globin, this is that protein, those 4 protein chains that we saw, the 2 alpha and the 2 beta subunits. The globin, well, it's a protein, so it's going to get broken down further. And like any protein, it's made of the same stuff, amino acids. So the hemoglobin is broken down into heme and globin. The globin is broken down further into amino acids. And those amino acids, what's going to happen? They're going to get recycled by your body to make more proteins. There's only 20 amino acids, and our body uses them to make any protein that we need. So these amino acids, they're going to get reused, maybe to make more hemoglobin, maybe to make any other protein. But let's look to see what happens to the heme. The heme is broken down further. It's broken down into iron and something called biliverdin, which is then converted to bilirubin. Let's go over and look at the iron first. The iron is going to be reused, is going to be recycled to make more erythrocytes. Now, some of it is stored in places like the liver or the red bone marrow. Some of it is immediately going to be reused to make more hemoglobin. Some of it is going to actually just get lost, maybe through menstruation in females, maybe through injury, but primarily iron is reused, recycled to make more hemoglobin. The biliverdin is converted to bilirubin. Bilirubin is actually a bile pigment. So bilirubin is taken up by the liver, converted to bile. Bile is then secreted into the small intestines, and it leaves the body in the feces. Now, if you're into kind of weird facts like I am, the bilirubin in the bile pigment is actually what gives feces its color. So if you ever start having really, really weird colored stools, that's often a sign. Your doctor will say, let's check your liver. Because if your liver is not properly pulling the bilirubin from your blood, if it's not properly releasing it into the bile and not properly releasing it into your intestines, then your feces will often be weird colors, white or gray. They will often call it clay colored. Now, let's look at this video. Now, again, there's not going to be sound. Unfortunately, it doesn't let me play the sound through a video like this. But what we're going to see here is this is a macrophage. Here are some red blood cells that are at the end of their life. And I'm going to let you see everything that we just talked about, even though you won't hear any of the sound. So there, it's split into the heme and the globin. Amino acids, they're going to go be recycled to make more proteins. The heme is split, and there's the iron to be recycled. Biliverdin, going to be converted to bilirubin. Bilirubin traveling in the blood gets taken up by the liver. Now it's part of the bile, secreted into the small intestine. Now some of it actually gets reabsorbed in the blood and the kidneys remove it, but most of it leaves the body in the feces. So let's go back real quick and look at this. This bilirubin, which is taken up by the liver and leaves the body as part of the bile in the feces. What happens if someone's going through liver failure? What is some common symptoms? Well, we just said often the feces will have weird color. But what else is really common with someone with liver failure? Jaundice. They get kind of this yellow tint to their skin, to their gums, to their eyes. What's happening there? Well, if the liver isn't functioning properly, a lot of times it won't pull that bilirubin out of the blood. And the bilirubin is what's building up in their blood and causing that weird yellow tint. In babies, especially premature babies, the liver is one of the last things to actually start functioning. So it's not uncommon for babies to have jaundice because their liver isn't yet pulling that bilirubin out of their blood just yet. All right, so we're going to stop there. At the next lecture, we're going to start talking about blood groups, and this is going to be important for both lecture and lab. And we're also going to have some lab talking about blood groups and blood typing. All right, so this is where the lecture is going to end today, and I will talk to you all again soon.